All right, so this is the nucleic acid worksheet, and we're going to start with question 1. Is this DNA or RNA? Of course, we're going to remember that in DNA, we have T's, whereas in RNA, you have U's for the bases. So this must be DNA, right? So this is DNA. Then the next question says, what's the complementary strand sequence? So you have to just be really patient. Remember that this is DNA is anti-parallel double helix. So we are just patiently gonna fill out without making a mistake the opposite strand. G, T, G. And this is 3' end. Right, and now if we know that this molecule, DNA molecule, contains start codon encoding beginning of the protein, let's find the start codon and figure out which strand is template, which one is coding. So I'm going to scan the strand, right, and I'm starting to look for, oh, here is ATG, right? So here's one possible start codon. I'm going to continue and see if there's any other ATGs. So as I scroll through, I don't see it. And then I can also monitor the other strand for ATG. I see AGT, not quite the same. And then notice something. This ATG is not really a start codon. Why is that? Right? Because it's in the wrong direction. So we never read DNA 3' to 5'. We always read DNA 5' to 3'. And so this one is not a start codon, not ATG, right? Because of the orientation. All right. So then if the start codon is found on the strand we added, let's label this strand. That's going to be coding strand. And then the other will be template strand. So it will be used as a template to create messenger RNA strands. So then let's write out, so now we're on number 4, write out messenger RNA that can be produced from this DNA label that ends. One question for you is, does the messenger RNA start with a start codon? The answer is, of course, not, right? Start codon tells us, actually, let me change color. Start codon tells us where translation is going to start. Messenger RNA has to start so 5' has to start before that's untranslated regions. All right, so we're going to start with, whoops, where we had, no, something didn't work quite right. Where we had A, it will be U. Where we had T, it will be A. And it's basically, if you notice, same sequence as we U, G, G, G. Oh, this is hard. You really have to pay attention. C-U-G, A-C-U-C-C-U-G-A-G. Oh, hope I didn't make a mistake, right? This would be mRNA. And so notice that the sequence of it, UAC, right, is complementary to the template strand, and it's very similar to the coding strand. TAC in the language of RNA is UAC. Okay. And then AUG is then going to be our start codon. That's going to encode, let's try a different color. That's going to encode methionine. And then for the rest of it, we're going to read 3 letters at a time and figure out what these codons encode, right? So then we need to go to the genetic code table. So here's my start coloring. AUG is the start codon. So the next codon is GUG. So I'm going to find, here's a G, then I need a U, which would be in here, and GUG is right there. So it's valine. encodes amino acid valine. So I just need to keep going, valine. And then if you just follow this, you should get valine, histidine, leucine, threonine, proline, and phenylalanine. F is also phenylalanine. All right, so that's our amino acid sequence, right? So let me just check I didn't make a mistake. I'll check the last one, GAG. So I'm looking for G, then go into where A's are, and whoops, that doesn't seem right. That's glutamic. Hold on, did I screw something up? 3 at a time. No, GAG. Now I have like doubts of everything else. So GAG, how about CCU? CCU is proline for sure. So GAG, sorry about GAG, should be E-glutamic. All right, oops. Glutamic. Okay. I'm not sure how that happened. All right, so we translated. We're going to label the and with specific terms. So that's N terminus to C terminus, right? And the protein will continue. And again, notice that the start codon is not where messenger RNA start. We still have untranslated regions. All right, so last one is what if we have a mutation? So if we change underline T to an A, right? So we're going to find that T. Let me highlight it. So this is the T that got underlined. So if that changes to A in the mutation, then the top strand is going to change it to a T, right? And that would change the last codon from GAG. So the mutation is going to be GAG to GTG. So is that going to create change in amino acid? So again, we're going to need to go to our table. Here's the GAG and GUG would be valine, right? So this would change glutamic acid to valine. So would that change create a problem? Glutamic acid, if you look at the amino acid sheet, has a carboxyl group and is negatively charged, right? And valine is nonpolar. So in terms of the question, how will that affect the protein? Could potentially can disrupt tertiary, possibly quaternary structure if that exists, and function, right? Because structure determines function. All right. Yeah, so that's the problem. So you have one extra practice problem. And so I encourage you to try to answer that on your own. And then on Canvas, I posted the answer sheet. All right, the last one we're going to try is going to be this diagram showing the models, right? And so the question here is, can we identify each process that's represented by a model, label boxes with specific names of the molecule? So the way you would approach this is, let's see what we have and what we're making. It looks like we start with DNA helix, right? There is double-stranded DNA, and we're producing two other DNAs. So this, the DNA to DNA, is DNA replication. Okay. And then on the diagram below, if you notice, we start with DNA, but our product here is single-stranded molecule. So most likely that's going to be transcription, producing a single-stranded molecule. Okay, and then let's just kind of start labeling. Right, so we have double-stranded DNA. That's a start. And then we're gonna have an enzyme that needs to unwind, right? And the primase and put down the primer. So this is gonna be, I'll put a note here, primase and helicase, the unwinding enzyme. Helicase can also be either this one, this right here, uh, or like if you label this one as helicase, this little donut thing, I'd accept that too, even though technically it's a slightly different enzyme. For students who've studied that in like micro, that's actually topoisomerase. For this class, just think we need to lay down the primer to be able to synthesize the molecule and unwind. We don't care about topoisomerase. Okay, then that starts with the molecules. Then let's think about how the synthesis is going here. So it looks like one strand has new nucleotides adding here, right? So that's going to be going 5' direction to 3'. That's how we synthesize DNA, right? And that would make this end 3', and the opposite end of it is 5'. Okay, so if this one is synthesized continuously, going 5' to 3', this is going to be the opposite, 5' to 3' going this way. And so it must be done in kind of fragments, right? So this would be the discontinuous replication. And so this will need to be primer, RNA, right? So down at the beginning here, and then the polymerase is adding new nucleotides in this direction. So 5' to 3' direction. So this would be new DNA. This is also new DNA, right? And the orientation, if I continue 3' to 5', kind of flipped, right? And this would be 3'. All right, what else do we need to label? So this enzyme here is going to be our DNA polymerase. And in terms of labeling strands, the top one is going to be lagging strand, whereas this one is going to be leading strand. Okay, so I think we got this one done. We switch color as red looks a little aggressive. Okay, so down below, let's look at transcription. So here we have an enzyme that's like represented by a big blob, right? That's going to be our RNA polymerase. Again, the reason we know this is transcription with RNA polymerase is because we start out with a double-stranded DNA. We're unwinding, and the product we're making is single-stranded, and one of the strands is not being read, right? And so the DNA strand that we are reading is going to be our template strand, right? And so we need to label it. So it would be this strand of DNA being read as template strand, while the strand not being read right here is going to be coding strand. Okay, then we're making new messenger RNA or new RNA, and it's going to be in the 5' to 3' direction, right? And that means the other end is going to be 3' and 5'. And on this side, we're going to have 5' and 3'. Okay, I think we'll label this one, right? So it's transcription because it produces from DNA template, it produces RNA. So I hope this was helpful. Good luck studying. Mm.